Welcome to the Bodhi Academy. My name is Shrutika and today let us continue with our chapter Molecular Basis of Inheritance. So today in our today's video what we are going to learn is the machinery of enzymes which are required for the which are required for the replication process. So last video we have seen what is replication and why it is called a semi-conservative model of replication. So today what we are going to learn is how this replication process happens in the DNA and which are the two processes which are related to the replication, that is transcription and translation, and what is the meaning of translation and transcription. So how the DNA replication happens in the double-stranded DNA. So there are various enzymes which are required for this process. The first enzyme is called as the DNA polymerase. So DNA dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of the DNA cannot be separated in its either length, the replication occurs within a small opening of the DNA helix referred as the replication fork. So a replication fork. So the DNA dependent DNA polymerase catalyzes polymerization only in one direction, that is through 5' to 3' direction, and this creates some additional complications at the replication fork. On one strand, the template with the polarity 3' to 5', the replication is continuous, while on the other strand, that is the 5' to 3', it is discontinuous, and this discontinuously synthesized fragments are later joined by enzyme ligase. So let us see what is a replication fork. So these are the two strands of DNA. So DNA helicase enzyme binds to the one strand of DNA and it helps in the unwinding of the DNA, wherein at the point of origin the replication fork is formed. This replication fork, then the DNA polymerase enzyme binds and it starts synthesizing new nucleotides, which helps in the formation of the new DNA. One strand is continuous and the other one is discontinuous, that is, it forms into small fragments, and each fragment is called as Okazaki fragments. And then after synthesizing the newly synthesized strands are then joined together by enzyme ligase, which helps in the joining of the nucleotide. So this is how the replication happens in the DNA. So now let us see what is the process of transcription. So transcription, the process of copying genetic information from one strand of the DNA into RNA, is termed as transcription. So what happens is, from the DNA, the genetic information is transferred into a single strand, that is the RNA, and is termed as transcription. So this is about the process of transcription. So the transcription process happens primarily, it has three regions in the DNA, that is the promoter, the structural gene, and the terminator. So this is the promoter region, wherein it is the site where the transcription starts. Then there is structural gene, and then there is a terminator site where once the template reaches the terminator site, then the new strand which is synthesized is released. So this is how the structural unit of transcription unit looks like inside the DNA. So let us see that in detail. So it is the process of copying genetic information from one strand of DNA into RNA. In transcription, only one segment of DNA and only one strand is copied in RNA. So the adenosine forms base pair with uracil instead of thymine. So transcription of DNA includes a promoter, the structural gene, and a terminator. So the strand that has polarity 3' to 5' acts as a template and is called as the template strand, and the other strand is called as the coding strand. So the promoter is located at the 5' end, and that binds the enzyme RNA polymerase to start the transcription process. So the sigma factor also helps in the initiation of transcription. So once the RNA polymerase binds, so it requires the sigma factor so that the initiation of the transcription process starts. So the The terminator is located at the 3' end of the coding strand and usually defined at the end of transcription, where the rho factor has to bind to the terminator side so that the termination of the transcription process happens in the DNA. So now let us see the process of translation. So what is translation? So in translation there are also 3 steps. So this is about the process of transcription, as in this is the initiation step wherein the DNA polymerase binds to the coding strand, and then to that the sigma factors bind, and then it starts the process of elongation, that is new RNA formation, that is no new nucleotide, complementary nucleotide are added and you get a new strand. So once the nucleotide reaches the terminator site of the DNA, what happens is the rho factor binds to the RNA polymerase, which helps in the release of the newly synthesized RNA strand. So this process happens in 3 steps, that is initiation, elongation and termination. That is called as the transcription, wherein from a DNA you get a RNA strand. So this is called as the process of transcription. Now we will see the process of translation. So let us see now what is translation process. So the translation is the process of polymerization of the amino acids to form a polypeptide. So the order of sequence in the amino acids are defined by the sequence of bases in the mRNA. Amino acids are joined by peptide bonds involving the following step, that is charging of the tRNA, formation of the peptide bonds between 2 charged tRNA, and the start codon is AUG, and mRNA has an additional sequence that are not translated called as untranslated region, and for initiation ribosome binds to the mRNA at the site of the codon, where the ribosome moves from the codon to codon along mRNA for elongation of the protein chain. At the end, release factors binds to the stop codon and the terminating the translation and release of the polypeptide from the ribosome. So basically translation is basically from the mRNA you get the protein sequences, that is you get the peptide bonds, which then further forms the protein which are required for the for synthesis of the amino acids. So this is translation process, wherein this is the ribosome, this is the smaller unit and this is the larger unit of the ribosomes, wherein this is your mRNA which is synthesized from the transcription process. So this has base pairs, wherein what will happen is your tRNA comes and binds, and then the ribosome, the mRNA will move. Again new tRNA will come and form the base pairs, again it will start moving. Hence it is moving, it will release the polypeptide chain of the nucleic acids, amino acids, which will further form the protein. So this is the process of translation, wherein from mRNA you get the proteins, and this process is called as translation. So in today's video what we have seen is what is translation process, what is transcription process, and we have seen how the DNA replication happens in double-stranded DNA. So thank you for watching today's video. Please don't forget to like, comment, share and subscribe to the Bodhi Academy.